For many years, evolutionary biology was held on the idea that most changes in proteins were invisible and did not impact the life of organisms. However, a new study published in Nature Ecology and Evolution argues that evolution may be filled with useful mutations that rarely leave a mark on genomes. This discovery provides a new framework for understanding how living beings change in a constantly changing world. If you like this content, don't forget to subscribe and like. Every support counts to continue sharing interesting information about biology and evolution. Now, let's explore how this new research challenges traditional ideas about evolution and mutations. The neutral theory of molecular evolution has dominated for years, suggesting that most genetic changes are neutral. This theory, though elegant, is based on the premise that beneficial mutations are extremely rare. However, the new study casts doubt on this assumption, suggesting useful mutations were more common than was thought, even if they did not look at time. The researchers analyzed more than 12,000 mutations that alter amino acids in 24 genes of different organisms. This comprehensive approach allowed observing the effect of each mutation in a controlled manner without theoretical assumptions. By measuring how each mutation influenced the organism, it was found that more than 1% showed beneficial effects. Although useful mutations are more frequent than expected, the genetic record among species still seems largely neutral. This raises the question of why, if useful mutation is common, they hardly ever leave a mark on evolutionary history. The answer could be in the changing nature of environments. Researchers suggest that environments change faster than assumed. A mutation that is beneficial today can cease to be so over time. This is known as antagonistic pleiotropy, where a mutation can have different effects in different contexts. Thus, many advantages are temporary and do not consolidate in the population. This continuous process of adjustment, called adaptive monitoring, means that adaptation occurs but is not fixed. Useful mutations appear and function for a time, but disappear before settling in the population. This phenomenon challenges the notion that evolution is a linear and constant process. From a broad perspective, genomes seem to accumulate changes without a clear direction. However, this pattern can be misleading. What is recorded in DNA is only the final result of an active evolutionary process that occurs in the short term and often does not consolidate. Adaptation remains active, but is constantly restarted in the face of new environmental challenges. This new theoretical framework relocates neutral theory into a broader context, suggesting that what seems to be lacking in selection could reflect an intense ephemeral adaptation. This implies that many natural populations may be less adjusted to their environment than was thought, although not for lack of evolutionary capacity. Evolution does not move towards a fixed goal, but responds continuously to a changing environment. This change of perspective raises new questions about how to interpret genomes and evolutionary history. Understanding evolution requires observing the ongoing process, not only the final result left in DNA.